Okay, ready or not, here we are. Another edition of Grumpy Science with two grumpy scientists, Mark Tommerdahl, Bob Dennis. Welcome back.
Yeah, and we just kind of realized we've done a lot of these already, and you guys aren't suffering through 45-minute hour lectures that we normally give our undergraduates, but
just to torture them, actually.
Yeah.
Not a lot of content, but they have paid their tuition, so
we thought we'd give a little recap of the previous videos, and that way newcomers could skip all those previous videos, and we could talk about sort of like what students like to do. They skip the first 3 weeks of class and hop in and
they'll
see what
check
we're
out
doing.
somebody else's notes, right? So this is the Cliff's Notes version. Can I say that? Yeah,
Cliff Notes version,
and
sure.
we don't edit, so there it is.
Yeah,
this is the Cliff's Notes version of Grumpy Science and a recap of what we've already discussed. So to begin with, we're grumpy.
And we probably just violated some kind of trademark mentioning Cliff Notes, but who knows? Okay, so here's the brain gauge.
People don't read anymore, so
yeah,
that's
true.
About it. That's why we're doing videos. We stopped publishing papers.
To do a quick recap, here's the brain gauge. It's a device that we spent 15 years shrinking down from, it lost 100 pounds in 15 years. And so we crammed a lot of stuff in here with the attitude that we could vibrate the fingertips extremely precisely. And the reason we do that is to activate places in the brain very precisely. And as we mentioned, basically the nerves in your fingers are connected to your brain. Now, some lawyers will argue with that, but we really do believe that there's significant proof from about 200 years of neuroscience.
But why believe that when somebody else who doesn't know anything can formulate an opinion? So we just recapped the last video. Okay, we'll leave it at that.
Yeah. And so what can you do with this? And basically you can deliver stimuli and deliver vibrations to the fingers and do certain tests. And in a nutshell, you can the various tests test different aspects of information processing in the brain. And it's a functional test. It's not anatomic, but it is using the tactile system. The tactile system has been, people since the 1950s or even earlier, I just only read the papers back that far, have been saying somatosensory testing would be the best way to test brain function. However, nobody could build anything that could deliver reproducible stimuli.
Available, right? They just weren't practical. They weren't available. So that even though theoretically and practically the best, well, not practically, but theoretically the best way to test brain function is using the somatosensory system. Mark went over that in detail a few videos ago, why that is true. And it turns out it is true, but people weren't doing it because they couldn't. They didn't have the tools to actually take the measures.
Yeah, and so even a simple test like reaction time is much more accurate with something like this than it is with anything else for a lot of reasons. Mm-hmm. And proof's in the pudding. If you do reaction time tests of visual versus auditory versus tactile, somatosensory is actually the slowest. Visual is the fastest. And if something happens, if you get a bang to the head or something like that, the fastest pathway, the visual system, really doesn't change nearly as much as somatosensory because you have what's called redundancy of function. And if you have lots of input to the eyes, it activates all these different areas, and that pathway gets there.
Your eyeballs connect to your brain at 8 or 10 different places, and maybe something somewhere is damaged or inhibited, whereas somewhere else just kind of picks up the slack. And there's really no way for you to tell with any kind of reliable precision that something either has or has not happened if you're looking at just sort of reactions to visual stimuli.
Yeah,
as far as I know, you can't do it.
Anyway, that's sort of the recap. And one of the things is it's a functional measure, not an anatomic measure. So what's the difference? So why don't we go to our media center? And
Mark, would you please enable the multimedia command and control center?
What's that top word that I'm about to pull down?
That one? That's a special word. You might use that.
It might even entail a special discount code, but you never know. We have hidden discount codes in our talks, not on this board, but on the previous. So anyway,
Mark, kudos on the penmanship.
Yeah,
almost as good as mine. We actually put black ink on the paws of a rat and had it walk up and down on there.
Yeah, well, if you go back to my second grade grade reports, I did fairly well on most subjects except a couple. One was penmanship, and one happened to do with socialization or something like that.
Yeah, other people,
the one
whatever. Who cares about other people? I'm a scientist. Get away from me.
I actually did very poor in reading for a while, but they put me in the worst reading group, and then they found out that I was reading the book upside down because I was bored.
Oh, well,
Yeah.
that explains a lot. Not going to say anymore.
Okay, so I wrote down the names of a whole bunch of different techniques you may have heard of. A lot of people ask us questions, and we actually
Let's go down the list just so people aren't
So we
squinting
want to
too
tell
much.
you what differences
Let's do
there
the
are.
list first, right? MRI. Everybody knows what that is.
Well, or do they?
Or do they, right?
Okay, people hear it all the
resonance
time.
Imaging.
And magnetic
MRI.
resonance imaging, which is anatomical. MEG, it's actually functional. PET. Now,
Positron emission
now the difference
tomography.
between these two,
Positrons.
difference between these two and PET, besides resolution, is that with PET you got to have a radioisotope.
Mm-hmm.
Okay.
Right.
So you got to actually inject something that's radioactive into your system and your bloodstream, and then it takes about 30, 45 minutes for that tracer to complete.
Favorite factoid about PET? P is for positron. Positron is antimatter. Antimatter really exists. It's not just Star Trek-y kind of stuff.
E is for emission, so it's emitting those
it's
radioisotopes and they're collecting them.
There you go.
All right, SPECT. That is also known as poor man's PET.
Single photon emission.
It's done a little different. X-rays. Everybody knows what X-ray is. We'll skip that. Ultrasound. That's what you do when somebody's pregnant. You go visualize them. But there's been a lot of advances in ultrasound technology, and people are improving resolution, and they're also doing this in brain stuff. By the way, we always talk about brain stuff. EEG. That's doesn't have very good spatial resolution.
Right?
Not very good spatial resolution, but very good temporal resolution. In other words, it can pick up changes in time. So it can be used as a functional measure.
Picking up electrical impulses that you can sort of almost pick up a little, like an antenna, surface electrodes on your skull, on your head, used very often.
Yeah. Optical. Now, optical imaging is a little different. Like you might shine a light on a surface, and neurons absorb light when they become active. It's all wavelength dependent, and to my knowledge, nobody's been really successful at this being non-invasive.
Yeah, basically you have to expose the brain, and if you have an extremely high dynamic range, I think is the correct term.
There are actually some papers using
the brain
this
function because it changes optically on its surface, right?
Yeah, there were actually some papers using this in the former Soviet Union before the wall fell down. Before they split up, where they made all sorts of claims about their optical imaging techniques, where they could just put a camera over somebody's head and read what's inside the head. You can.
Well, as they say in Russia, you do not watch TV. TV watches you.
Yeah. I'll skip down to CAT scan, or computerized automated tomography. You know what that is?
CAT scan?
Yeah.
It's when you're looking at cats.
Okay, cool.
Okay. So.
Now, what is fMRI, and why did they put it down here? Well, I didn't want to put F in front of MRI, but fMRI uses the exact same technology as MRI. Difference is MRI is anatomic and only takes a snapshot of your anatomy. All right? That's the easy explanation. Functional MRI. What you do with functional MRI is you generally have to deliver a stimulus. Now, what we've been talking about is tactile stimuli. You can deliver tactile stimuli in an fMRI, and we actually are parts of a lot of experiments that do that. We build stimulators for those.
So an fMRI is kind of looking, I think it's mostly metabolic changes that can be imaged.
Yeah, we can do a
with
short.
An MRI.
It's mostly blood flow.
Yeah, it's blood flow. And so, like, if you do something that stimulates your brain in a certain way, you can actually image increase or decrease in blood flow to different regions of the brain. So it's not a super fine-grained high resolution image. It's more kind of,
Well, the difference is now
color.
People would argue with me, but mostly all you can really see with fMRI is activation. Same with EEG, activation. You really can't measure inhibition.
That's true. Yeah. So don't interpret what I said earlier as meaning inhibition so much as, a change in blood flow doesn't necessarily mean inhibition, and in fact, inhibition can be active, right? So it could correlate with an increased blood flow, right? So
yeah.
So there you go.
And there's a couple other technologies that use the same hardware, One's MRS, and that is actually what that does is it measures neurotransmitters, sort of the brain chemicals. And so you can measure things like GABA, which is the main inhibitory neurotransmitter, DTI. And what that's good for is looking at the integrity of white matter. Okay, so that's sort of like a very high resolution anatomical study of is your white matter still broken up. And so some of these studies have been used to show that white matter integrity is worse when reaction time variability goes up. Now, reaction time variability is a quick measure, and it's easy to do with sensory perception. And it gives you, but it doesn't necessarily, if you do poorly on it, it doesn't mean your white matter integrity is bad. But it's a functional measure. That's the difference.
Right. So I think we're at almost 11 minutes, Mark, which means we are almost out of time. And I just finished my beer before this.
Right. So basically, all we wanted to do is mention that these are mostly measures that look at things like the functional types of measures that you can do with those. Usually involve blood perfusion, blood flow, where is blood flowing to the brain, what parts of the brain are damaged.
So
this is stuff
contrast agents in, but usually then that's sort of a proxy for something like blood flow.
And there's a lot of really good research being done with those different methods for very specific things. But we get questions about how is a brain gauge different from all of these methods.
That's the question that Mark is going to answer in the next 30 seconds. How is brain gauge different from all these other things you can pay thousands of dollars for?
Well, one way it's different is that it's portable, it's quick, and you can measure different functions from one to another. We actually have one measure that we haven't rolled out yet, but one measure that we have does correlate with some of the MRI or MRS measures. Some measures actually correlate to some of the DTI measures. Some actually correlate with some of the EEG measures, and we've done studies with all of those. Some measures actually correlate with what you can get with fMRI. The difference is the brain gauge is a lot cheaper, and it won't do mapping of the brain anatomically.
We don't anatomically map the brain. We functionally map out what it's doing and what it's not doing and how well it's doing it. It's a very different thing. So you shouldn't expect an image of the brain showing, oh, there's a lesion here or something like that.
It's a little bit like a blood pressure cuff. You can get a blood pressure cuff and check your heart rate and decide what your blood pressure and your heart rate is, and that's a functional measure. Or you can go do MRI. You can do some kind of imaging to see if there's any tears on the heart.
Right.
So
the sorts of things we are interested in terms of brain function are not generally related to changes in the anatomy or the structure that would be easy or in principle easy to image as an anatomical image. We're not really interested in those things. It's sort of like changing the software in your computer doesn't change the physical computer itself. It changes the way that it's functioning, right? So what you're looking at with brain gauge is a functional map of what's going on in a functioning brain rather than an anatomic or a function correlated with anatomy image. That is not what we do.
Okay, we should stop now.
Okay.
All right.
Thank you very much.